Good afternoon, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. Our webinar today is a Precision Oncology Roundtable, and we will ask the experts about NGS-based tumor profiling. And our sponsor is PurionDX. Our panelists today are Dr. Rakesh Nagarajan of PurionDX, Dr. Gregory Sangalis of the Dartmouth-Hitchcock Medical Center and Norris Cotton Cancer Center, Dr. Anthony Magliocco of the Moffitt Cancer Center, and Dr. Ravindra Kolhe of Augusta University. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after all the presentations have concluded. First up is Dr. Rakesh Nagarajan, Founder and Chief Biomedical Informatics Officer at PurionDX. Dr. Nagarajan, please go ahead.
Great. Thanks so much, Christy. I'm going to go ahead and jump right into my slide deck. Our first slide here just talks about our one-space solution portfolio. I won't spend more than 30 seconds on this, but essentially we have a set of products and services that enable a clinical lab to perform clinical next-generation sequencing on their own. And the way we do this is through three of the services we'll talk about today, the CGW, the interpretation services, and validation services that's offered through professional services, and I won't really talk about gateway lab services today. This slide really just briefly talks about the cancer care continuum with respect to laboratory testing, which is where I'll focus down at the bottom, where the trend is to move from very focused single marker testing to more and more comprehensive panels, and we'll talk about one of those comprehensive panels today. That assay today that we'll focus on is the Illumina TruSight Tumor 170 assay, and the content of that has about 170 genes, both DNA and RNA-based, including allowing for low DNA input as low as 40 nanograms. It does cover SNVs, indels, copy number variants with respect to amplification, and gene fusions using using the RNA-seq results. This assay can obviously be run on the NextSeq and HiSeq instruments. This slide really talks about the composition of the assay. So again, broad solid tumor base of the major cancer types, obviously, that are shown here, and again, varying detection of both DNA and RNA targets, including copy number variants. I want to spend just a minute on assay validation and the services we provide there, which includes that for TST-170. So the first step in that process is to really design the experimental approach to help decide numbers and types of samples, how they get run on different sequencing runs, determining clinical performance targets and assay claims, selecting then samples for analytical and diagnostic validation, and then doing both components of that validation with the partner laboratory that includes typically methods-based validation of using reference controls, and then doing a variety of calculations, including specificity, sensitivity, positive predictive value, lower limit of detection, and diagnostic validation components as well. I want to now then focus on the CGW, or the clinical genomicist workspace workflow, and how we've integrated the TST-170 into that workflow. So namely, as you know, within CGW we can do order entry through either manual order entry or integration through HL7 or API-based access, as well as bulk accessioning. The process then from sequencing to report is much the same within the system, except that some of the details are different for the TST-170. One is that we use the TST-170 app that's available either through base-based integration or through a local deployment, and all of this is seamless to the laboratory, whether they use the base-based solution or the local solution. And then we also have our own modules that I'll describe validation for that calculate tumor mutation burden and microsatellite instability. This slide really then says that once variants are identified using the TST170 app and have been validated through a validation process, whether with us or the lab has performed it independently, the CGW then allows you to view variants of interest that are filtered by quality, and based on the validation filters that were set up during the initial process, you can also view sample quality metrics both for DNA and RNA, and get down to read level data, as well as a variety of other knowledge-based components that I'll talk about in future slides. At this point, I want to kind of drill down into the initial validation we performed both for TMB and MSI. Let's start with TMB. So TMB, as many of you know, it measures the quantity of mutations that a tumor contains, and that's being used to predict whether a patient will benefit from immunotherapies. It's typically calculated as the mutations per megabase. Folks have shown that you can calculate this using whole exome sequencing, but now there are a variety of folks that have also shown that you can do this in targeted panels. And that's what we aim to do with TMB calculation in TST170. So this slide really shows analysis that we've done both using whole exome data, as well as then limiting that, the variance in the whole exome to the TST170 capture region. And we use TCGA data sets to calculate tumor mutation burden in greater than 10,000 different TCGA samples that represent around 30 tumor types. In this particular calculation, only non-synonymous somatic variants were considered. When we limit then the capture region to the TST170, there's still a high level of concordance, both in value and relative levels across tumor types. That is, lower tumor types known to have low tumor mutation burden in the literature continue to have that in whether we analyze using whole exome or TST170. This is further than shown here, where through direct correlation and concordance, we can show that either all mutations that's shown on the right or non-synonymous mutations, if you only use them, are both highly correlated to whole exome-based analysis after limitation of those variants within the TST170 capture region. This last slide then on the TMB validation now looks at TMB calculation and using that to differentiate responders from non-responders. And again, using the TST170 capture region, we can show that both in non-small cell lung cancer and melanoma, there's differentiation that we can get, and that differentiation is held true whether we use the whole exome or, again, only the TST170. Moving on to microsatellite instability. So, again, many of you know microsatellites and the number of repeats within those microsatellites may be altered in tumorigenesis, and this is typically caused by mutations in the mismatch repair genes or through epigenetic inactivation of those genes. And obviously the status of MSI, which is typically today which today is really determined by PCR of about 5 loci, has significant in cancer prognosis and implications for therapeutic response. This slide really then shows our initial validation analysis, and again, we took TCGA results and samples where the MSI was known in those TCGA samples, and again, we calculated microsatellite instability across thousands of loci that had microsatellites within the whole exome, and then limited to the TST170 capture region. And again, you can see that MSI status is very highly correlated to the number of MSI events, which is what's plotted on the y-axis, as compared to the MSI status that's determined during PCR, whether it's microsatellite stable, low, or high. And I want to kind of jump into the actual visualization and reporting in the CGW. So once variants are called through the TST170 app and classified and reviewed, our knowledge base creates an automated classification for these variants. which includes a wide range of therapeutic, prognostic, diagnostic, and risk information and options, as well as clinical trial matching. I want to spend just a minute on our knowledge base itself. So the knowledge base is comprised of, at the very lowest level, publicly available sources, so human genome builds, gene mRNA protein models, monoallele frequency database information, the published literature itself. There are sources that we then clean and curate, normalize to a standardized disease oncology that includes data from Cosmic TCGA and laboratories who release their results. We have highly curated information from from sources such as NCCN and ASCO and FDA-approved labels, which is germane for the somatic cancer setting, as well as clinical trials. And finally, we have a proprietary knowledge base of shared interpretations that are shared across our partners, of which there are greater than 50 today. And that really forms a very powerful network of sharing interpretations that have then been written by medical directors in a particular disease context, and those interpretations then infer across the network in a future case for medical directors whether that variant was found in the same or different disease context. This slide just shows an example of one of the many report templates we have that laboratories can choose from. And again, you can report out all the different variant types. You can report out TMB and MSI results. In pages that are not shown here, there's just the first page. You can also see per variant individual trials, targeted therapies that you can then edit fully within the system to sign out the report. I want to end by talking about our interpretation services, which is the third service then of the four that we're covering today. So we truly believe that the right mix of technology, including the platform and human medical expertise, will ultimately help in genomic medicine. And we've provided that technology and platform through the CGW, which again provides automated classification of variants, provides a knowledge base for therapeutic, prognostic, diagnostic, and risk options, and can do clinical trial matching based on molecular findings. The human medical expertise that we now offer through variant scientist, medical director, and including sign-out services, should you need that and want that, allows for assessment of variant classification and interpretation, and updating that information based on the newest literature, drafting clinical interpretations and reclassifying variants based on information that you gather from the system, evaluating pathway-based associations and covariant associations to create cohesive interpretation, and then doing clinical trial matching that is based on pathway crosstalk as well as client-specific SOPs. With that, before I hand it over to Christy, I just want to summarize that within the CGW, you get a complete workflow from order entry to a signed-out report, and we do that for the TST-170 as well as a variety of other assays. And then we provide interpretation services at the end of that process should you want or need that service for either scale reasons or personnel reasons. Christy, I now hand it over to you.
Thank you, Dr. Nagarajan. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Gregory Tsangalis, Director of the Laboratory for Clinical Genomics and Advanced Technology at the Dartmouth-Hitchcock Medical Center and Norris Cotton Cancer Center, and Professor of Pathology and Laboratory Medicine at the Audrey and Theodore Geisel School of Medicine at Dartmouth. Dr. Tsangalis, please go ahead.
Thank you. Thanks, Christy, and thanks, Rakesh, for the opportunity to share some thoughts with everyone today. Hello, everyone. What I'd like to do is just kind of give you an overview of some of the things we think about in the lab when we're talking about next-generation sequencing with respect to the different panels we run and some of the things that we're thinking about implementing. I think everybody's familiar with this first slide and the real power of the technology that we've seen coming out of next-gen sequencing or massively parallel sequencing, and the advantages that this technology affords us in the lab with respect to being able to analyze really complex human diseases like human cancer, and that's the focus of the talk today. I think before we talk about some of these applications, we have to really think about the added value that the laboratory is able to bring to the table with respect to patient management and patient outcomes, given that the tests that we're providing are not so much used for diagnostics, but very much used for prognosis and selection of different therapies. And before we start talking about some of the things that we implement in the lab here, I just want to call everyone's attention to this really spectacular guideline paper that came out just this year in the Journal of Molecular Diagnostics that I think everybody should read before you even think about validations of a next generation sequencing assay or panel. And so with that in mind, I just wanted to show you a couple of slides about what we're doing. Because if you remember back in the day, or still in some labs, we were running singlet assays for certain genes, certain mutations, and the disease becomes much more complex than that. And I think everybody appreciates that, and we've appreciated that for a while, because we talk about cancer as a clonal disease that's shown in this top panel here, with select mutations being acquired by cells as they progress to become a malignancy and then metastatic disease. And that's really a way underestimation of what really happens, and I think we're appreciating that now with all the tumor cell heterogeneity. But if you add to the biology of the disease what's happening in the pharmaceutical side or the treatment side of the disease, it becomes even more complex, because every one of these abnormalities that these cells go through is now, or seems to be now, being targeted by a different therapeutic option for these patients. And so that becomes really the big impetus for us to start looking at technologies that allow us to generate a lot more information than what we could with some of the other more traditional techniques that we're also familiar with. In this slide, I'll just highlight one pathway. This happens to be the pathway for EGFR, just to show you and highlight the point that there are multiple genes with all kinds of variants in them that act either in a singlet or in conjunction with each other, and maybe in conjunction with other pathways and so on, that we used to be able to do one at a time, but it's just become way too complex to do that. And so that's kind of the focus of what most of us have been doing with these panels of genes and applications in the oncology space. So what I like to think about when we're talking about expanding the gene, the panels are listed here. And so the title of my talk was, how many genes do you want to test for, and so on. It just seems like right when we thought we were doing a great job and we have a nice panel or panels in place, the request comes in for more genes, or there's a new application and we have to add genes, and so on. And so we talk about what's the clinical utility of this. Are we really going to impact patient care? Will we have cancer-specific panels in the lab as there are commercially available, or will we have pan-cancer panels that we can use across the board? And again, there are a number that are commercially available, including the 170 that people are talking about today. What types of variants are we going to detect? We thought we were doing a great job with the SNVs, and then the CNVs came about, and the fusions, and all of these things now become important. So you have to kind of figure out what it is you really want to be doing and detecting in the lab. For me, one of the biggest questions is the next one, and that's what about the workflow and the lab logistics, including informatics. This is something that I think we way underestimated what it would take to set this up in the laboratory, and we've been doing this for almost 4 years now. What about the platforms and the chemistry? There are certain instruments from different vendors, depending on the assay you're going to set up and the type of chemistry you want to use, and that becomes important for you to evaluate. Money for equipment and capital funding for automation is a big deal, where we didn't think we were going to need this a few years ago. We certainly are looking for as much automation as possible now. I think training techs and the extent of the learning curve is something to keep in mind. the turnaround time that your oncologists are expecting and what you can really deliver is another issue. And then cost and reimbursement, although last year and this year reimbursement seems to be happening a little bit at a time now. So we should be good there, I hope, in this next year. So let me just show you two cases that we had recently. One was a 57-year-old female that had non-small cell lung cancer with an ALK-EML4 fusion that made her eligible for treatment with crizotinib. And after 4 months, the disease progressed, and as we sequenced her, we noticed that she also had, or the tumor had, a P53 mutation that was associated with a really poor prognosis. But in addition to that, she also had a non-synonymous point mutation in the ALK gene that's associated with resistance to all of the ALK inhibitors. And there's only one publication of this in the literature that we could find at the time anyway. The second case is a 50-year-old male with rectal cancer, had follow-up, developed some pelvic mets, was treated with FOLFIRI, which is a chemotherapeutic regimen, and cetuximab, which is an anti-EGFR antibody therapy, and had great response, despite the fact that he had a BRAF mutation. And so BRAF mutations have been associated with no response to the anti-EGFR therapies. Well, it turns out that in this case, he didn't have the typical V600E mutation. He had a D594G mutation, which again was reported once in the literature as being favorable with response to cetuximab. And so I show you these two cases because they really highlight the need to be able to do good sequencing across genes and gene regions with a level of coverage that's going to allow us to detect a lot of different variants. Some of them we may not know what they mean right now, and I think that's the importance of our databases that we're all putting together. But there are others that we would have missed or you'll miss if you're only looking in particular exons of certain genes. And I think these two cases and other people have many more that kind of highlight that need and the importance that will mean for these patients and their management strategies. I think this is my last slide, just showing you the team that we have in place here at Dartmouth to be able to do all of this, and we continue, like everybody else, to ride the ride, so to speak. And so I'll turn this back over to you, Christy. Thanks.
Thank you, Dr. Sangalis. We want to remind participants again that if you have a question, please type it into the Q&A box, and we will conduct the Q&A once all the presentations have concluded. Next up is Dr. Anthony Magliocco, Senior Member and Chair of the Department of Anatomic Pathology at Moffitt Cancer Center, Executive Director of Esoteric Laboratory Services and the Morsani Molecular Diagnostic Laboratory, and Scientific Director of the Moffitt Tissue Core. Dr. Magliocco, please go ahead.
Great. Thank you for inviting me to share our experiences at the Moffitt Cancer Center. I'm pleased to be here today. So at Moffitt Cancer Center, one of the key features of our center is a focus on precision medicine and personalized oncology. And at Moffitt, like other places, we believe that precision medicine is really defined by having a correct diagnosis, about assigning a correct treatment, doing it in a timely way. But also, we're seeing more and more that we need to adjust it because we have multiple lines of therapy now. We have trials that are linked to evolution, and it's becoming more and more complex. We're also understanding that each individual solid tumor is unique, and the type of progression and response to therapy and surgery, et cetera, may also be unique. So we need the tools to manage that and deal with that in this complex environment. Now, fortunately, at Moffitt, we were lucky to recruit Dr. Howard McLeod, seen here standing proudly in the center of this image, and he's led our personalized medicine service. And this is a comprehensive service that's really helped us in the molecular lab, because this service provides personalized medicine consultation to oncologists. So anytime a large panel is ordered or there's a question about what should be done next, his team will work with the oncologist to pick the right panel and help interpret the data. For particularly challenging cases, there's a molecular tumor board where these are discussed on a biweekly basis. Dr. McLeod's created a training program where he's training multiple fellows, actually from a diversity of backgrounds, including molecular pathology and pharmacology and others, that we hope will go forward in the future to other medical centers across the country and help found new personalized medicine services as well. There's opportunities in pathway development. In Moffitt, there are prescribed pathways for each treatment type, and we believe this is the foundation to evidence-based medicine, and it's also very useful for our payers, that if we can convince our payers that we're following a rational approach and a predictable path of treatment, they're more likely to compensate us not only for the testing, but also for the treatment we choose. And more to that end, there's a service to match patients to clinical trials that may be open at Moffitt and beyond, and also to potential off-label opportunities for patients with rare conditions or unusual tumor profiles. Finally, the personalized medicine service does work with payers and has been very successful in obtaining reimbursement and coverage for off-label use in other applications. So kudos to Dr. McLeod. Now, in terms of expanding our panel, we've been running NextGen for a number of years with smaller focus panels. I believe we've ran over 8,000 NextGen panels over the last 3 or 4 years, and the time came that we needed a bigger panel. So we lean towards TST-170 because we've worked with Illumina for a number of years, and we feel that they're one of the leaders in this particular field. And so our instrument base was Illumina-based instruments, so we were drawn towards their reagent profiles. We believe that there's a very broad implementation base of users and that many other users will also use this panel. We've previously used their off-the-shelf TST-26, their heme panel, and other solutions from them, and we were impressed with the quality of those products in the past, and that gave us more confidence moving towards TST-170. They also have an opportunity for custom panels, and one of the negative aspects with Illumina is they came from the research world, and they're really just cutting their teeth now in the medical diagnostic world. And only a few of their products have gone through FDA process, et cetera, but they are learning this quickly and are certainly poised to enter the medical market with greater potential. When we looked at Illumina, we looked at other panels as well, including Archer. And actually, we feel that Archer has a really great panel that we may be implementing into our liquid biopsy service. In terms of Illumina, we also looked at their 500 gene fusion panel, which covers 508 gene translocations. Now, at Moffitt, we have many, many demands from our different tumor services, so we're trying to satisfy demands from lung, endocrine services, brain, and a variety of other tumor sites. And essentially, we do a survey of what they need and try to match to provide them with an optimal result. We felt that the 170 was the best available result for our particular needs at Moffitt. We're also planning to launch that along with the 500 gene fusion panel, so we'll have a comprehensive set. And down the road, we are looking at more focused panels, perhaps in the 50 to 70 gene range for liquid biopsy purposes, CSF, and cystic fluid analysis. Now, one of the values of TST-170 that we've discussed a little bit earlier is that it's got comprehensive coverage of a lot of cancer-related variants, and this panel was constructed with the intention of actionable targets in mind. So it's really focused towards actionability. So some of the mutations that may be more defined diagnosis may not necessarily be covered in the panel, and this has caused some consternation for renal services and perhaps neural services and so on. But in terms of treatment selection, it's a very good panel in my opinion. It also has DNA and RNA aspects, which is really optimal for identifying fusion and splice variants. And there's a unified workflow that works fairly well. The other thing that's quite good about this panel is that it works with fairly low input, as low as 40 nanograms of DNA and RNA, and that it can detect as low as a 5%, so the analytical sensitivity is down to 5% mutant allele frequency, which we think is quite good. We've covered the workflow before, so essentially the data comes off the instrument, it goes into base space, which is housed by Illumina. It's a HIPAA-compliant cloud, and some processing is done there, and then it goes into the Purion data workflow, where it comes out as a report at the end. We heard a little bit about that at the beginning. We've worked with Purion from the beginning with our launch of our first NextGen panel several years ago, and we found them very convenient to work with and very adaptable for our specific needs, helping us prepare reports in an efficient way and an accurate way for our particular clients and in internal use at Moffitt. So we've been fairly happy with this process and the ability to modify this, adjust filters, and test the workflow. We're in the middle of this, and we're hoping this panel will actually go live in the next week or two. So we've spent several months running through the recommendations of the diagnostic profiling article from the journal Molecular Diagnostics, and have run about 80 of these assays. And we're seeing very, very good results, which I'll show you as we go through there. We've determined that single nucleotide variants we can detect down to 5%. Amplifications with 10 or more copies can be detected in tumors with 30% tumor load, and fusions and splice variants can be detected with greater than 5 copies in 1 nanogram of RNA. So we believe that this meets the performance characteristics for solid tumors and the type of samples that we typically see. It may not be adequate for liquid biopsies, but that's a story for another day. The sensitivity and specificity of the assay is that for small variants, there's a greater than 95% sensitivity and specificity at the 5% when coverage is greater than 250x. For fusion and splice, there's a greater than 95 specificity for greater than 5 copies of mutant RNA per nanogram. And for amplicons, again, a greater than 95% specificity and sensitivity for variants at a copy number equivalent to greater than 2.2. So really excellent performance across all of these categories that need to be covered for solid tumors. And we've been busy going through this, and these stars represent that we've detected these in multiple samples in cross-platforms and in our controlled specimens. So to date, we've been very happy with the performance of the assay, and our intent is to bring it live. The other aspect that I didn't really cover in this is the mutational load, which I think will be a greater need as well, greater demand coming from the immunotherapeutic services, as we now see indications based on microsatellite instability across tumor sites. So we're really expecting a dramatic increase in demand for these types of assays in the near future. So I'd like to thank Dr. Terry Boyle, who led the project in getting this assay validated in-house, and all of the assistance that Purion and Illumina has given us to get us to this point where hopefully we'll launch it in the next couple of weeks. So that concludes my component. Thank you.
Thank you, Dr. Magliocco. And as a final reminder to webinar participants, if you have a question, you still have time to put it into the Q&A box, so please go ahead. Next up is Dr. Ravindra Kolhe, Associate Professor of Pathology, Associate Director of the Residency Program, Medical Director of the Cytogenetics Laboratory, and Director of the Georgia Esoteric and Molecular Laboratory at Augusta University. Dr. Kolhe, please go ahead.
Thank you, Christy. First, I would like to thank Rakesh and his team for giving us the opportunity to share our experience with TST-170. We are a state medical institute academic place, and we have a tripartite mission of clinical diagnostics, education, and translational research support for on-campus. So first, I would like to begin with why we ended up choosing TST-170. As most of the lab, we have either a DNA-based or RNA-based panel in our lab. The DNA-based panel is mostly for the heme malignancies, and the RNA-based panel is the sarcoma fusion panel on RNA-seq. But in a clear setting with any type of assay, irrespective of its NGS or even a single gene assays, a lot of careful consideration has to go through the content of the panel or the genes. Especially, you want to make sure that you have a good evidence-based markers selected in the panel, so you're set for next 3 to 5 years as a panel for clinical testing. And for me personally, I think it's very important that these panels are compatible with low quality of DNA and RNA, because as a surgical pathologist, I usually look at these slides, identify the region of interest on these H&E slides, and isolate the nucleic acids. And most of the time in the past, they have not been compatible with the assays we had on the NGS platform. So when Illumina presented to us the content of the TST-170 app in March of, I think, 2016, it was really helpful to go through, and I was very happy to see what the content was. I mean, the assessment of fusions, splice variants, insertions, deletions, SNVs, amplification in one single assay for looking at both DNA and RNA. It definitely creates efficiencies in sample usage, time, and cost for us. So I'm going to share a couple of slides with our experience with TST-170, initially as a site for the beta testing and later as a validation. As of today, we have tested around 89 different samples on the TST-170 app. I think the 8 of those were for the beta testing. We're one of the 3 sites, and as you can see, I think most of this data is publicly available as a white paper, which can be downloaded there. But in our experience, we had a good inter-site performance for all the variation SNVs and different variants Illumina has on the panel. But apart from that, all the different samples we have tested have worked very well. I think Tony also in the previous talk mentioned about their experience, and or has been pretty similar to that. But this is the usual workflow for most, if not all, of our samples. All our samples were FFP samples. We had a microdissection for DNA and RNA isolation. We use QIAGEN kits for the DNA and RNA isolation, which has worked very well in the past for NGS assays. The library prep was pretty much from the box. We used NextSeq for sequencing, and the initial variant call was on the base space on the TST-170 app, and we are collaborating with PredientDX to create a template and architecture for reporting of all these variants. And I think you have consistently seen that in the talk. So the other added advantage was we were able to get the workflow done in such a way that we could run 2 runs in one given week. We would start the first run on Monday and finish the sequencing by Thursday, the overnight sequencing on Wednesday, and start the second run on the Wednesday of the same week. So this helped us to have 2 runs on a NextSeq in one given week, which really helped us to finish the validation in a given time. And as I initially mentioned, we run 8 samples in one run on a NextSeq. So some of the results from the study and the validation, in our experience, this is relatively simple protocol as compared to what we have done with bigger assays or whole exome sequencing. The reagents were pretty integrated as a part of the kit, and I was really happy to see a built-in informatics provided by Illumina. I mean, one of the things which I like to coin is it was really an NGS in a kit, because, or NGS in a box, because it helped us to have a solid library prep along with the different things which were needed for running the assay, as well as the initial bioinformatics for calling up the variants. We had a strong operator on the site, as well as run repeatability as a part of the beta testing. The sensitivity and the call concordance was observed across all sites, runs, and operators. And in our ongoing validation, I think we just started another run this morning, but we have run around 81 sample, and we had a pretty accurate variant detection, and which was comparable to all our validation samples to orthogonal methods. As I initially mentioned, we had all our samples as an FFPE. They extended from punch biopsies to surgical resection. These are the common questions I get asked for these talks, so I'm just going to be forth with them. The RNA we used was 89 nanogram in total, with the input volume of 8.5 microliters, somewhere between 8 to 10 nanogram per microliter. And the DNA we used was around 120 nanogram total, with input volume of 12 microliters with 10 nanogram per microliter. So this has been our experience with overall the TST-170 kit. In summary, we are very happy with the TST-170 app. It's a pretty comprehensive DNA RNA panel. I really feel it's a very cost-effective way to investigate the majority of the malignancies, as the coverage is pretty decent. One of the things which I'm trying to do with Peridian is create templates for reports, and I think at least our reports will have 3 layers. The first layer will be the tumor-specific information, or what I like to call as TSI. So this layer of information will be very specific for particular tumor, which will be based on guidelines for diagnosis, prognosis, and therapeutics. For example, if you take colon cancer, it will have the CAP, ASCO, AM guidelines for the RAS-RAF pathway detection in colon cancer. The second layer will be the expanded therapeutic targets, which would be for the precision and personalized oncology. These are the targets which will be detected based on the different variants we'll identify in the panel. And the third layer of information will be a combination of the MSI and TNB for immuno-oncology folks. So this, I think, overall will be a pretty comprehensive reporting structure. I think we will be able to generate with the help of Peridian DX. And this is my last slide. Thank you.
Thank you, Dr. Kolhe. Before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And we'll now start the Q&A. The first question is for all the panelists, and we'll start with Greg. How often are patients treated off-label based on the identification of unorthodox mutations that are actionable in tumor types other than the tumor sequenced? And Greg, specifically for you, did the sequencing findings that you had affect the treatment plans for the patients that you spoke about?
Yeah, so that's a great question, thanks. And the answer is yes to the management strategy changing for the patients that I spoke about. Overall, I'm not sure that I can give a percentage of patients that get treated off-label, but I think one of the things that I found that really surprised me was how often oncologists are willing to use therapeutics off-label if somebody's failed first or second or sometimes third-line therapy. So it's not something that happens infrequently.
Tony, do you want to weigh in?
Yes, the off-label use is frequently considered when patients have exhausted conventional lines of therapy. So they generally follow, the standard treatment, and then when the tumor has progressed, these types of assays are run, and opportunities, the first choice is to assign to a clinical trial. But if a clinical trial is not available, then an off-label use is considered. And often, as we said, that's been approved by the payers in our environment. And because it is a carefully managed strategy through the personal oncology service that really, makes an informed decision as to what's in the best interest of the patient.
And Ravi?
The answer is yes, and to add to Tony's information, I think we have had this experience with heme malignancies. When you've exhausted your current treatment post-trial, I think the data from the assays like this have definitely helped us to deal with the payers for pre-authorization.
And Rakesh?
Yeah, I don't have anything to add to this particular question.
Okay, thank you. The next question is also for all our panelists, and we'll start with Ravi on this. How do you return results back to your patients, and what recommendations are usually made?
So as a lab, we usually report it back to the ordering, either the oncologist or hematopathologist, whoever is the ordering physician. But also on campus, we have a cancer genetics clinics where the patients are on one-on-one. They have the opportunity to discuss their reports in depth, and the pathologist or somebody from the lab services is available for them to discuss the variants or what's the importance of the variants and things like that. So we have a protocol in place to deliver the reports either to the oncologist or to the patients.
Tony?
We use the Pirion system, and it reports back into our electronic medical record, which is Cerner. Currently, we only provide services for our own in-hospital patients, so that data is available to any of the physicians that are available at Moffitt, and also goes to the personalized medicine service as well, if for additional overview. And like I said, that service is sometimes triggered for complex cases to work with the teams to help make further treatment decisions.
And Greg?
So we report directly to the attending oncologists, and our reports are pretty basic right now, and that's why we're looking at Pirion DX for that service. But I can say we also have a molecular tumor board that reviews difficult cases, and a lot of discussions come up now at our routine tumor boards.
Rakesh, the next question is for you. Your data for whole exome sequencing versus TST-170 seems to suggest that we can do away with standalone MSI testing. Would you agree with that?
Yes, so that's an interesting question. This is preliminary analysis on TCGA results. We have some follow-up analyses where we also looked at MSI testing either by IHC or PCR and calculated specificity, sensitivity, positive predictive value, negative predictive value, and again, got almost perfect results there. And, whether you can do away with MSI by IHC or PCR, I think we'll need to do a larger number of validation studies with individual laboratories in order to really make that determination on a laboratory-by-laboratory basis. Remember, this is not an analysis that's an FDA-approved device. It really needs to be validated within individual laboratories. But yes, I think the evidence is very good to replace the current approach to MSI with this more comprehensive assay, and appropriate diagnostic samples will need to be validated within each lab to do that.
The next question is again for all of you, and we'll start with Tony on this one. Do you think there have been enough studies that prove the clinical utility of comprehensive cancer genomic profiling to improve the quality and quantity of the life of cancer patients?
In terms of collecting clinical utility data, that is extraordinarily challenging, in terms of looking at, does this data change treatment decision-making? Does actually changing the decision have an impact on outcome? What's the cost-benefit ratio of all of this? And I think that will take a significant amount of time to really, prove the clinical utility. However, there's no question there's a significant clinical value to these tests, and we see, significant evidence being able to identify patients for approved therapies like anti-BRAF treatments in melanoma, in tyrosine kinase inhibitors, and now for immuno-oncology treatments, that these tests clearly have value in identifying patients for approved usage of approved therapeutics. So I don't doubt that there will be more evidence accumulating and that these tests will become standard of care down the road.
Ravi, can you weigh in?
I mean, I fully agree with Tony, and as he said, definitely it adds value to the clinical decision making, but the cost benefit has to be determined locally within the institutes or within your practice, and so on.
And Greg?
So I think, we've all had patients that have had absolutely remarkable responses to the therapeutics they were getting, and then we have other patients that have had incremental at best responses to whatever the therapeutics were, even if they're directed by genomic profiling. I think one of the things as a field we have to be really careful of is how we report these things out and what we're reporting as far as the mutations go and so on. And we need to be much, much more specific, even if it means re-educating everybody on what the mutations are, because not all BRAF mutations are the same, not all EGFR mutations are the same, but lots of trials and studies only require a positive or negative for one of those genes and so on. And so I think we have a lot of more data to collect to answer some of these questions. but we have to be better at collecting the right data.
And Rakesh, would you like to add anything?
Yeah, I think this is the chicken or the egg thing, and this has been an academic debate for about 6 years now with, greater and greater adoption. That's a groundswell from both patients and physicians, as well as organizations such as those on the call who are, believing in the scientific and medical merits of more comprehensive profiling. I think there have been a few papers now that have been published talking about clinical utility of a more comprehensive assay than very targeted, FDA-approved companion diagnostics. And as both a clinical cancer research paper and an onco-target paper, where additional trials are identified, a greater number of mutations are identified, and even more basic things like deletions in exon 19 of EGFR that are outside the scope of very limited assays are identified through these manuscripts. So, I think the evidence is growing, and I think there are also initiatives that payers are starting to believe in where they're willing to pay in advance, collect the data, and then evaluate the clinical utility together.
Great, thank you. Rakesh, does Purion offer the NGS TST170 related workflow as a validated diagnostic label test as you describe in your presentation? And is this service FDA cleared?
Right, so that's a great question. So, as all of you know, this highly complex modality is offered as LDTs by individual laboratories. We support that activity by partnering with our laboratories where we validate both the wet and the dry component in order to create a complete LDT. So this is by no means FDA cleared to be very specific, and the assay itself will be validated individually with each laboratory as we've done in the past.
Our next question is for everybody, and we'll start with Greg on this one. Did anyone validate the TST170 panel with the advertised low input amount of 40 nanograms for FFPE samples? And similarly, what is the lowest DNA and RNA input that you find acceptable for attempting to run a patient specimen? And do you ever issue qualified reports with lower inputs?
So I'm going to defer the question because we've not started to validate the assay yet.
Okay, Tony?
I think we've been pushing it down to the limits, and we've done 50. We haven't really pushed it below that, and we felt it's been performing quite well at the low end. In fact, there's some speculation whether it might even be able to perform at lower than that, but we can't say for sure. But at the low end, we have been impressed by the result.
And Ravi?
The answer is yes. We, as a part of the beta testing, and a few of our samples and the validation had a lower DNA, and we have gone below to 40 nanograms.
And the next question is again for all of you. What would be the time scale for a clinical molecular lab to set up this kind of testing in terms of going from zero sequencing to providing meaningful validated sequencing data? Greg, do you want to start with this one?
Wow. I really think it's going to depend on the experience the people in the lab have, and I think you're looking at anywhere from a few months to 7 or 8 months.
Ravi?
Yeah, I think I agree with Greg, and it all depends on what your past experience has been on the NGS, the platform. It's a pretty comprehensive panel, but as I said, the workflow is pretty decent. So in our experience, we had some past NGS, clinical NGS work done, so we were able to get this thing up and running within 3 to 4 months. So that's if that helps.
Tony?
Yeah, it depends where you're starting from. If you're starting from an empty lab and you're trying to train scientists and technologists and select equipment, get that going, you're going to have to take at least a year to get everything running. If you're running other NGS-type panels, you're pushing it 3 or 4 months. You're doing really well. It's because of the number of runs that are required that you can only do so many on your equipment, so it depends what your throughput is, how many runs you can get out of it on a weekly basis, and so on. So these large panels do take a significant amount of effort. Remember, there's both equipment, training, there's wet lab, and then there's dry lab as well, and the dry lab takes time as well. So a lot of aspects to them, and it's a big undertaking.
And Rakesh?
Yeah, I think the other panelists have covered this well, but what we've seen across laboratories that we've partnered with, and some have never done NGS before, and others already had the equipment in place. It can definitely vary from 8 to 12 weeks on smaller assays outside of TST-170 to up to a year, as some of the panelists mentioned. They're waiting to get the equipment, learning how to use the equipment from one or more vendors, getting automation in place. As Tony mentioned, getting the IS team involved to do integration with LIS or EMR, getting the lab ready to do validation, designing the validation, running through the validation, coordinating that with the bioinformatics team to get the dry part done, deciding what the report is going to look like, what content it'll have. Lots of decisions to be made and many tests to herd.
Great. The next one is again for all of you. Are any of you planning to use the TST-170 panel for germline testing? Tony, if you could take this first.
Yes, we are. We're looking at that after we get this launched, whether we can run normal along with that to help us determine what the variants mean. We also know there's indications now for PARP inhibitors and so on the DNA repair enzymes, and that might also play a role in the MSI side. So we are looking at that, although we don't have a plan in place yet how to actually make it happen.
Ravi?
Same answer as Tony. I think we first want to get for the tumor part, and then once we settle and up and running, then we're going to revisit with validation for the germline.
Greg?
So yes, I agree with what was said so far, and I'll tell you that we, like most of the people listening, have kind of shied away from the germline for a long time with respect to cancer. And I suspect that now with the new applications for sequencing with respect to tumor mutation burden, MSI, looking at BRCA1 and 2 mutations for PARP inhibitor eligibility and so on, we can't avoid that anymore. And so as a group, we have to kind of decide how best to move that forward. And it would be great if someone else could write a guideline paper.
And Rakesh?
Yeah, what we've seen with our customers is when you start to get it to an assay of this size, it really behooves you to start thinking about doing germline. This one's probably right on the edge, but once you get to definitely 400 or 500 gene and beyond, having the germline paired non-malignant sample is vital to making rapid decisions on somatic variants. That's the first piece of it. The second is Greg mentioned understanding the heritable component starts becoming important, and folks are asking for that as well.
And the next question is again for all of you. Rakesh, why don't you take this one first? How do you deal with requests for testing for additional genes that are not included in the TST-170 panel?
Yeah, I'm only going to pass this to the 3 lab directors, given that we don't do testing directly. I'm going to let them answer that. Okay, Greg?
I think that if we'll review and see what the genes are and what the utility of mutations in those genes are, and if it's a valid request, we would send it out for a panel that included those.
Ravi?
Yeah, I think same answer as Greg. It is a pretty comprehensive panel. I would be very surprised that if there's anything in addition to that we would need to do. But again, we have been doing all this thing in the past is evaluate what the request is. If it's an evidence-based and helps the patient, we'll eventually end up sending it out.
And Tony?
Yeah, agree. We'll send it out if it's indicated and they need it. We've already had some requests for additional genes from the GU service, so we're looking at how to manage that. But we'll send it out if there's a medical need for it.
And Rakesh, will PurionDX generate a population-based database of variants with allele frequencies that can be shared with the research community? And are novel variants being submitted to public databases?
Yeah, great question. So, we're already creating that database and have created with respect to our, data sharing network that I described as part of the knowledge base, and that allows in the clinical workflow medical directors to review variants and their prior frequencies across tumor types, as well as how those variants were classified in previous cases, whether in the current patient's cancer or not. And Greg and Tony and Ravi can review that information, as can all medical directors as they review and sign the cases out. Now, the data, for research purposes, across the network, we don't have the ability to really release that information. That information in the clinical context is allowed per, our contracting with individual laboratories, but we would need appropriate laboratory consent, patient consent, or waiver of consent in order to release those data for research purposes.
Great, thank you very much, and that's all the time we have for today. We'd like to thank Rakesh Nagarajan, Gregory Sangalis, Anthony Magliocco, Ravindra Kolhe, and our sponsor, PerionDX. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.